Welcome to Shaping Science, a collection of videos from the voices that are shaping science today. I'm Alejandra Manjarres from Drug Discovery News, and today we're joined by Dr. Michael Dunphy from Walsh University. Analytical science plays a critical role in advancing pharmaceutical research, from characterizing new drug candidates to enabling safer, more targeted therapies. Dr. Michael Dunphy's research focuses on using analytical chemistry to control, measure, and translate highly reactive chemical species into real-world biomedical solutions. As Vice President for Academic Affairs, he has also led the expansion of research and analytical capabilities at Walsh University that support industry collaboration and hands-on student training. In this episode, we'll explore how analytical instrumentation makes this work possible. So, without any further ado, let's get into it. Hi, Michael. How are you doing?
I'm doing well, Alejandra. Thank you. Appreciate you putting me on here today for a little chat about chemistry.
Thank you so much for joining us today. To begin with, you've helped expand Walsh University's research and industry partnerships over the years. What have you done to build those connections, and how does Walsh now support pharmaceutical development?
It's a great question. So since really when I was on faculty back in the 80s, we were working at building partnerships with industry here locally and regionally, as well as companies that could support what we're doing. So many years ago, I was fortunate enough to meet Heather Jeswa from Shimadzu, and we very quickly built up a great relationship, and she was really instrumental in helping me acquire our first HPLC, which is back, I think it was back in the 90s. And we began the process of a long-term relationship. So over the years, we've been working here at Walsh University to have these relationships set up so that as we are growing, the companies who could partner with us would grow with us. We'd be able to get our students involved in research projects. We'd help our faculty do research. And up to now, because of the Spark grant that we were able to acquire in 2020, which built out fundamentally a $2 million suite of instruments, we're now positioned beautifully going forward to do much higher levels of research and be able to work with Shimadzu strategically.
Yeah, no, that sounds great. Well, now let's dive into your research.
Yeah.
Your work focuses on using analytical chemistry to create real-world biomedical solutions. Walk us through how decisions in the lab translate into potential drug candidates.
Yeah, so there's a lot there, Alejandra. From the beginning, when a compound is first synthesized, whether it's a larger molecule or a smaller molecule, it's important to develop two things. One is a way to measure it in biological fluids to determine its concentration as a function of dose. And secondly, to determine how it's metabolized. So for example, HPLC mass spec or GC mass spec would be a great way to determine metabolic patterns. And secondly, when you're doing initially dose response studies, you need to know how to measure it in blood. So chemists normally will develop these procedures early in the process. And if then the compound goes to clinical trials, then you're also going to be doing pharmacokinetic profiling, looking at how the compound behaves when you dose it, whether it's oral, IV, or whatever the dosing mechanism happens to be. So you can then understand how the half-life is, how the compound behaves in humans. And then eventually, as the material becomes common, if it goes to clinical trials, makes it out into the world, then physicians will want to do, likely on some of these compounds, therapeutic drug monitoring. So as they're dosing real patients, they want to see how the compound's behaving in their blood. And in some cases, we develop methods to do emergency toxicology, because if someone overdoses on something, end up in an ER, an emergency room, then you should be able to, on the back end of that, determine its presence and its concentration, both in plasma and in urine. So there's a broad range of uses for analytical procedures from chemists in the lifespan of a compound. And certainly on the front end, when all that research is going on, it's super important to have a system to measure the concentration of these materials in human fluids, for sure.
Yeah, sure. Yeah. One of these highly reactive molecules your work focuses on is nitric oxide, right? Mm-hmm. Could you describe what you've been working on and challenges you've overcome along the way?
Sure. So we've had the good fortune, one of my former students, Dr. Joe Bauer, when he was, this is back in the 90s at the Cleveland Clinic, he determined a way to attach nitric oxide, which is NO gas, to cyanocobalamin, well, actually it was hydroxycobalamin, a vitamin B12 derivative. And in doing so, was able to create a compound that cancer cells couldn't differentiate from normal B12, and so it'd be taken into very actively metabolizing cancer cells, and it would release NO gas once it had exposure to lysosomes. NO gas, when it's inside of a cell in high concentration, can be cytotoxic, and we found that this stuff actually can kill cancer cells. It doesn't have hardly any effect on normal cells because the uptake of B12 in non-cancerous cells is pretty small. So over the years, we did the same thing. We made the molecule around, Joe reached out to me somewhere around 2008, to develop an HPLC procedure to measure NOCBL, both on the synthetic side and also on the human dosing side. At the time, we were dosing dogs, actually, but it was looking at how do you measure it in plasma. And as a consequence of that, I had a bunch of my honor students work on these projects. And so our students here got the amazing experience of being able to develop from scratch these analytical procedures and then see the benefit on the back end when they were able to present what they learned and was able to help Joe with what he was doing. At the time, we were working towards a veterinary market for the compound first, and then eventually go to humans. And so NOCBL is just a great example of taking a natural product, modifying it slightly, and coming up with a pharmacologically active agent that's efficacious. So right now we're working on a modified version of that because the patent ran out. Now we're looking at a variation on that to get it out into the market. It's very expensive to do clinical trials and to do all this work. So we have to raise money, and sometimes it's grants and sometimes it's private investors. But the whole thing, top to bottom, is awesome for students to be involved with. And we used Shimadzu HPLCs to do the work.
Okay, that sounds exciting.
Yeah.
How are the findings translating to real-world solutions?
What happens is that the findings give us the ability to determine what dose should we use, how quickly does it come out in the urine, what's the mechanism of action, because we were looking at how do we understand how this material actually binds to cancer cells quickly and translate into nitric oxide doses being delivered internally. So all the studies we did, both biological and chemical studies, gave us information to determine, okay, what do we need to do with this? And we found, for example, that it was very pH dependent. If the pH dropped below 5, the NO gas was released immediately. So you couldn't give it orally because stomach acid would cause it to decompose so quickly it wouldn't work. So it has to be given subcutaneously or IV. Well, we determined that through our studies. So there's a whole range of things you'll learn when you start to really look at the details. So ultimately, we can say to a physician or to an oncologist, here's how you dose it, here's what you can expect, here's the side effects, if any, and here's what we know will be the likely results because of all the studies we have done. And ultimately, if you're going to go to the FDA and say, hey, we want this to be in a clinical trial, you have to come with a lot of evidence. Otherwise, they won't approve it. So lots of work is done on the analytical side to prepare for that, for sure.
Thank you, Michael. Now, how have analytical techniques and instruments evolved over the years, and how has that expanded what you're able to study?
Yeah, that's a great question. So they have companies like Agilent, Shimadzu, Thermo Fisher. They, of course, have large research facilities where they are always evolving their instruments. So they focus on how to make it simpler to use, more sensitive, easier to maintain, and able to do more with less. And so what's happened over the years is, for example, in the world of LC mass spec or GC mass spec, they've been able to create these triple quad systems that have 3 quadrupoles back to back on the end where the molecules are being ionized, which increases sensitivity amazingly. You can get in sometimes as low as, sub picogram levels on some compounds. So you can get more sensitive, which lets you detect the materials in much, much lower quantities and much smaller samples. The technology has also gotten better in terms of the way instruments are set up. We now have auto sampling systems that are just amazingly good. When I remember those first came out, they had little challenges with them. They've now evolved to be sort of standard. The instruments are more durable. They've built them to be able to tolerate the conditions that they're used under to last longer, and the maintenance has been made a little bit easier. So I think it's been great to see that in any of the areas, whether it's infrared spectroscopy or UV-Vis or chromatography. The companies have done a superb job of making these instruments just better, easier to use. Sometimes the expense goes up as a consequence of that, but you end up getting a better product. And so I think what's neat is that the instrument industry works very hard to be connected to the universities around the country and around the globe for that matter, because the more they work collaboratively, I think it's a win-win for both sides of that equation. So we've been delighted to work with Shimadzu. They've been amazing. And we're looking forward to another Spark grant to get more of their instruments, including triple quads, to increase our capabilities and give our students as much modern instrumentation and applications as we possibly can. So it's very exciting for sure.
Yeah, yeah. Well, and talking about the students, with your long career in education, how has hands-on experience with analytical instruments impact the way students learn and grow as scientists?
Oh yeah, it's the best way to do it. When students are able to get hands-on experience with high-quality instruments, a couple of things happen that I've seen it when I was teaching labs a lot. They get very excited about being able to sort of, we call it drive the instrument, so they can set it up, calibrate it, learning about calibration is a huge part of this, then do their analyses and see that things don't always go the way you think they're going to go. It can be complicated. They learn the instrument itself so that when they graduate, many of these students will go off into industry, and now they're competent on a LC mass spec or a GC mass spec or an infrared microscope or a, let's say, for example, a GPC. They've used it. They know what it is. They're easy to train once they go out into the world. Also, they appreciate the challenge of what it takes to be able to get good data. And that's an important part of doing these things. So when we got away from the whole recipe lab stuff years ago. Our labs are designed where we say, okay, we have this problem. How do we approach it? And they come up with strategies and which instruments they might use. How do you measure something? Which gives them the ability to learn really what happens in actual science when you're talking about developing something or trying to solve a complicated problem. So having the hands-on experience with the instruments lets them gain confidence in being able to approach these problems in a more direct way. That's one. And then second's real short. They're just fun to play with. So they actually love to be in the lab because they're doing something that is interesting to them, as opposed to just simply, following a recipe lab is supposed to get some result. When you work with these instruments, you're not exactly sure what you're going to get. So there's a lot of mystery in that. And then as they see results starting to go the right way, because they're making tweaks on the front end, it's exciting for them. It's just fun.
Yeah. Yeah, sure. Yeah, awesome. Thank you so much, Michael, for joining us today. It was a pleasure talking with you.
Well, thank you very much. My pleasure, and enjoy. I wish you all the best as you do what you do.
Yeah. Okay, thank you.
Take care. Bye-bye now.
To learn more about analytical science, please click the link in the description. And thank you for watching. Together, we are shaping science.